Hello, welcome back to Astronomy 305, Life in the Universe. I decided that I'm going to start the audio lectures on this module C6, Origin and Evolution of the Life on Earth. It's possible you attended the lecture before we had the extended spring break and saw some of this material in lecture, but since it will be covered on exam 3, I thought that I would start over from the beginning. So welcome back, and here is the first audio lecture for 305, and it again is the origin and evolution of life on Earth. Our learning objectives are searching for life and its origins. We'll review when life began on Earth and what that early life looked like. And at the end of the lecture, we'll discuss where it may have begun. And then the origin, we'll talk about our hypotheses on how life actually may have begun. And this is pretty nebulous thinking because there's not a whole lot of data to constrain the ideas, but they're pretty sound nonetheless. They provide an excellent framework for future research in this area. We will talk a little bit about if life could have migrated to Earth, but really that's going to be saved for one of the videos that we have in this module. We're going to talk about the evolution of life, the major events, and that's going to include the evolution from prokaryotic to eukaryotic cells, and the rise of oxygen is going to be critical in that step. We'll talk about impacts and extinction, not in great detail, but we will talk about something called the KT event, which is responsible for the period where the dinosaurs became extinct. And we'll talk about the continuing threat of asteroids and meteorites. And we'll end with human evolution, which is very interesting. We're sort of progressing off the curve of Darwinism, and so we'll spend some time talking about that. Now, we have 3 lines of evidence for early life on Earth. The first one here is definitely the most substantial, and there are entities called stromatolites. They're built up from colonies of bacteria that seasonally create layers of minerals, and they survive in these rock sort of formations that you see in the lower left. So they're called living stromatolites because, in fact, they are still colonized by bacteria. But there are ancient stromatolites, of course, which are residual leftover fossils of these colonies. And using radiometric dating, they date back to about 3.5 billion years ago. And if you remember your timeline on the Earth and Sun and solar system formed about 4.6 billion years ago. And the period of bombardment ended about 3.8 billion years ago. So it wasn't very long, on the order of 300 million years, that we seem to have colonies of bacteria on the Earth. The second 2 are not as solid. The second one's intermediate solid. These are fossils that are, again, mineral imprints of living creatures. And they date into rocks that go 3.2 to 2.5 billion years into the past. And so these results, again, are not completely without ambiguity, but the dating of the rocks is pretty solid, and the fact that they appear as fossils is quite solid too in their phyla structure. The last one is a little bit more subtle. This is through carbon ratio, isotope ratio dating. If you remember, there's carbon, which has 6 protons, and the most common element or form of carbon is carbon-12, which has 12 nuclear particles, 6 protons, and 6 neutrons. There are other isotopes, meaning there's a different number of neutrons, and so there's 13 nuclear particles in carbon-13, 6 protons, thus making it carbon, and 7 neutrons. So in this case, it turns out that living organisms have smaller C13 to C12 ratios than does natural inorganic matter. So the cosmic abundance of the ratio of carbon-13 to carbon-12 is a certain ratio, and then life tends to favor carbon-12, and so therefore has a smaller amount of carbon-13 incorporated into its biomass. And so there are regions of metamorphic rock that definitely show the carbon-13 to carbon-12 ratio that is suggestive that a living organism was squished and metamorphosized into the rock and was deposited there. Also, there's some small particles or small rocks that are called zircons, which is, it's not in the lecture here, but there are zircons from Australia, and in fact, some of these zircons date also way back to about 3.7 billion years ago, and they have sort of the carbon-13 to carbon-12 ratio suggestive that the carbon was processed through living matter. Okay, so we talked about our DNA in the previous lectures, and how we have genes and have miles and miles and miles, millions of miles of genetic code in our bodies. And so if you lay out the entire genome, which is to say you look at every single gene laid out and study its code sequence, you can match different entities, different animals to each other, or fungi to various different slime molds, or this kind of thing. And so you look at the percentage of overlap, and the closer percentage of overlap, it's believed that those that they're closer relatives in the timeline of the evolution of living creatures on the Earth. And so from that, starting in the 90s, they developed this thing called the Tree of Life. The Tree of Life is basically a proximity graph, and so you can see that eukarya are all in proximity to one another and seem to have had some kind of breaking off further in the past from archaea, and they seem to cluster together from a common ancestor. But the archaea and the eukarya, if you go down that final branch to the common ancestor, heading toward that branch, you can see that they had a common ancestor together, and that in fact bacteria may have had a common ancestor as well. So this is based upon, again, percent overlaps of DNA and genetic code. And this really does argue that all genetic codes on the planet are related to one another. And then a corollary of that would be that we would have a common ancestor. Okay, so in the 60s, people began to get very interested in these questions about how life may have originated. Scientific thinking had progressed far enough that people didn't continue to think about spontaneous generation of larvae and heaps of garbage and things like that, or in dead matter, like dead animals and dead people. And they started to really think about this from a biochemistry point of view. And so Harold Urey formulated an experiment with his graduate student Miller. And they did this experiment where they made 3 flasks, and they tried to mimic the process by which organic material on the planet may have been gone from just organic goop to, say, the basic building blocks of life. So their hypothesis was that life probably formed in tide pools near the oceans, so therefore the organic material could get concentrated in water, that the environment of the atmosphere was methane, ammonia, and hydrogen, which turns out to be an incorrect assumption, because there was a lot of nitrogen and sulfur as well that they didn't understand at the time. But anyway, they mimic what they thought was the early Earth atmosphere. And then you need energy to drive chemical reactions, so they assumed that was from electrical discharge from lightning that would happen in storms and whatnot. And so what they did was they mixed the goop into flask 1 with the air, and they let the water and the organic material sit in that flask for a while. and they moved it to flask 2 after it had time to mix, and they gave it a discharge of electricity in order to provide some input energy. And then what they did was then they moved it to the 3rd flask where they let it cool and condense, and then of course measure the constituents, and lo and behold, they got a ton of organic molecules. And so many of the organic molecules they got were amino acids, which are critical for building proteins in the human body and actually are some of the codons in your DNA. So that was a spectacular confirmation that you could, in fact, develop the building blocks for life just from natural processes. Things have gotten much more sophisticated than that. We've evolved into understanding the early atmosphere chemistry. We evolved in terms of understanding that there are other sites and locations where life could have formed, and that there are other forms of energy that could have been used, say chemical energy down in these thermal events that are deep down in the ocean, where there is no light other than there's a lot of heat from the thermal events. One of the things that's nice about that hypothesis is that you are safe from sterilizing impacts on the Earth. Okay, so how do we make the transition from regular chemistry to, say, biology and reproductive biochemistry? Now that is the key question, okay? And so when we look at the early Earth, if we follow the Uri Miller experiment, we've got a bunch of free-floating amino acids. Many of those are the bases for your DNA and RNA, the U, the G, the C, and the T molecules. And so the idea here is you have to recall that these molecules favor certain pairs. And since that C likes to bond with G and A likes to bond with U, at least in RNA, that's uracil, but thymine, T, is the one for DNA. But nonetheless, you have C and G that bonds with each other, and U and A that bond with each other. So you start out on the left there with your organic soup of all these free-floating amino acids. Then it turns out that they bond in these chains that follow the CG and the AU bonding pattern. And from there, you may have a lot of these long chains, and then you may actually find that you can get duplication, sort of going over to the 3rd panel from the left, where you can see where an RNA chain may actually duplicate itself through weak bonding and basically have a, in a sense, mirror copy of itself, not an exact copy of itself. But then if we go to the final panel on the right, you can see that the complementary copy then might make a complementary copy of itself, which would have the identical chain of amino acids that the original amino acid had. So that it would be a form of duplication. And so it turns out that this RNA world, or this RNA model, is a pretty good idea. Now, the thing is that, going to the next slide, we want some kind of way to catalyze this process, to concentrate these amino acids that once they're in these chains, that they're sort of like held together, pushed together, so that they sort of get involved in a rapid chemistry process here. And so in life, we have protein, which is involved in this process of moving the RNA around and holding it down and making it reproduce. And so we didn't have that in this RNA world, but people have hypothesized that something like clay would be very good. So, you know, you're in this watery organic solution with these RNA chains, or bases, as you might call them, and then the water sort of evaporates, it gets goopy in the clay, and then the clay can actually hold them together and help the process of the reproduction of the complementary pairs. So it could solve this idea that you don't have something like proteins to serve as an enzyme. It's good for concentrating the organic materials, and it turns out that people now surmise that, in fact, this could have played a very important role, and in fact, They've done laboratory experiments, and in fact, it has held up as a viable hypothesis. Now, once you start making a bunch of these RNA chains, you do wonder whether they're going to get broken apart by um, competing reactions that are taking place, and these competing reactions could A, use the resources, and B, break up anything that formed before it could make its complementary pair, and then its complementary pair reproduce its original um, strand. So it turns out that if you have lipids and amino acids, they tend to form little shells. So amino acids are, your basic little molecules like we've been talking about, the things that RNA themselves are made up out of. And so if you have cooling water, they tend to form into these little spheres. And so in the figure A down there on the right, the bluish figure, you can see are some pictures of some microscopic spheres that are made from amino acids. The other things that can form shells are lipids. They can form closed membranes. Now, if you've been keeping up with the COVID-19 information, you probably have learned now that the COVID-19 virus has these little spicules sticking out of it, and the sphere which surrounds it is a membrane of lipids. And inside is the DNA or the RNA, actually. So the virus is really exactly what we're talking about. Now, this is why soap is so effective at destroying the COVID-19, because it is a lipid membrane. And when you put it in contact with long molecules of soap, they basically just tear the thing apart. And therefore, then you've exposed the RNA and broken up all the spicules. And so the whole virus structure is destroyed. So that's why the soap and water works really well. And interestingly, we're talking about a life form which is upturning the world that has a shell, a membrane made of lipids. It's kind of interesting timing for this lecture, but I don't mean to have humor about that. Okay, so these shells have very great properties. First of all, they can grow and shrink by adding more, and they're very flexible in terms of size. They have surface tension, so if they somehow are being pushed apart or pulled apart, it turns out that they can split into daughter things, like you see soap bubbles doing and things like that. And then it turns out that they have some porous behavior properties that allow some molecules to pass through them and block other molecules out. Now, that's a very important part of it. And the other thing which is important is they can store electric charges on their surface, which is energy, which, as you saw from Uri Miller, is a form by which you can drive some chemistry. Now, let's say you get some RNA inside one of these bubbles, and the RNA amino acids can move back and forth through the membranes, but many of the other molecules that would compete for those amino acids in other chemistry are not allowed in. So now you've got concentrated ability without competition. And so this is sort of your Darwinian unequal reproductive success. Now you've got a situation where RNA is free to do what it wants without being in competition or being destroyed. And it has some energy, and it has the flexibility of that these things can split and make more, and therefore it can grow in number. So this is really a spectacular hypothesis that this kind of thing could have happened. Okay, moving on to this transition, going a little bit deeper, we can see that we may have had this RNA world of protein and molecules, that they may have been concentrated in these pre-cells or lipid and amino acid bubbles. But we know you've got to have water, and water is one of the molecules that can easily pass through these, because water is the transport medium of all of these various types of organic chemistry processes. Okay, so the upper right is just basically showing you how the protein ties together and facilitates or catalyzes this process in the modern day world. And then, if there was a point in time where the protein got involved in the process, we might have something like we have down in the lower right corner, where the RNA does have an enzyme, and that this is a very isolated and protected environment that is really very efficient at making complementary strands of RNA. Okay, so we're going to have a series of a few slides here. We're going to go through 5 steps that are in the images here. This is a slide we'll discuss steps 1 and 2. Step 1 we've talked a little bit about. It was experimented in the lab as the Uri Miller Experiment. It's basically you've got tide pools, you've got energy in the form of sunlight, you've got energy in the form of lightning, you've got an atmosphere, you've got organic material being deposited on the planet through comets and solar system debris in the early formation epoch of the Earth, and you even have the black smokers you can see down there in the left figure, where you've got energy input, and lots of biochemistry could take place in those environments. Okay, moving on to number 2, you may have developed an RNA world that use clay as a method for concentrating and catalyzing this process. So we talked about that in quite detail, where you get an original, then you get a complementary copy, which I'm calling a sister, and then you get the original copy, and bingo, you've got self-replication going on in these in this clay muck. Okay, now moving on to number 3, which is the middle diagram. Then you may have these membranes form out of the lipids, or you may have them form out of other amino acids. They can store charge, they can duplicate and grow, they can be different sizes, and they have osmosis properties so that only certain molecules can pass in and out. You limit the biochemistry competition, you concentrate the materials that are needed for RNA to do its gig of making its complementary copy, and then going through the second process of duplicating itself. So if that happened, all that happened, then eventually you're going to get to the process where this is very efficient through unequal reproductive success, in the sense that this process just naturally is occurring, and we might have what we call a bona fide primitive cell. This would be number 4, but that primitive cell would not be with DNA as we know it, it would be with RNA. And maybe at this point in time, some proteins got involved in the process, because replacing the need for clay or something like that. And we really have what's a very efficient mechanism for preserving and concentrating this RNA genome to reproduce itself. Finally, number 5, after a lot of time, a lot of evolution, a lot of environmental stresses and unequal reproductive success, you get to the point where the RNA has become more robust. It's developed in a DNA molecule, and now proteins are involved, and you really have your true first prokaryotic cells. You have things that have a full DNA complement in them, and that have membrane around them, and maybe even specialized structures that help them actually move, which that means that they would start to have response to stimulus, which was one of our definitions of life. And you can truly get what might be the first simple biological systems. Okay, now let's move on to the timeline of the development of life. Take a little pause here. If you have a cup of coffee or a cup of water, take a sip. I'm going to do that. Okay, so we're back to our timeline down at the bottom of the slide. These are eons. In the slide, the time scale is in billions of years. This goes from 0 at the right, the present day, to 4.6 at the left, which was the formation of the solar system. I think each tick is 100 million years, so quite a long time. Okay, so if you remember right, we started on the left there in the Hadean eon, and the Earth formed. We had sterilizing impacts, bombardment of the solar system and Earth. And then we started getting formation of a lithosphere around 4 billion years ago, and then we talked about those carbon isotopic evidence that life may have formed as early as that. We got zircons, which have the C13 to C12 ratio. That would be suggestive that early life existed at those times. And then, of course, moving to 3.5 billion years ago in the Archean period, we have the first fossils that were found in rocks, and they date all the way back to about 3.5 billion years ago. Now, somewhere along the line, we developed materials like cyanobacteria. We developed anaerobic metabolism. And in this anaerobic metabolism, one of the things that would happen, these would be photoautotrophs in the sense that they used photosynthesis, they used light for their energy, and they used carbon dioxide for their carbon source. And by consuming carbon dioxide from the atmosphere and in the water, they would process it, they would release oxygen, and so basically to get their carbon, they break up the carbon dioxide molecule, and so 2 oxygens are released for every carbon atom that they process, and lo and behold, you're releasing a ton of oxygen into the atmosphere slowly over hundreds of millions of years. It's believed that this process began to kick in around 2.5 billion years ago and really was, going gangbusters around 2 billion years ago. So oxygen really began to accumulate in the Earth's atmosphere somewhere around 2 billion years ago. And it turns out that this is about the same time that prokaryotic cells evolved into eukaryotic cells. Okay, and if you remember right, the eukaryotic cells are more complex. They have a double membrane, but most importantly, they have organelles. So the chloroplasts, which were what process the sunlight into energy in the cyanobacteria and the plants and whatnot, would get incorporated as an organelle into the eukaryotic cells. Also, mitochondria showed up, which do the ATP cycle. And these are specifically aerobic photosynthetic cells. They don't necessarily exhale carbon dioxide or exhale oxygen by processing carbon dioxide. They actually need to process oxygen in order to carry their energy and do their ATP cycle. Now, the interesting thing about this is that for most life, the high oxygen concentration was actually poison. It was toxic. It was too reactive. It would, kind of tear the cells apart. And so those that were able to, process and survive through higher oxygen concentrations survived unequal reproductive success. And eventually we have a aerobic photosynthesis dominating the planet. That's not to say that anaerobic metabolism still doesn't exist. And in fact, your body is capable of doing both aerobic and anaerobic metabolism. And that's left over from this period in the early Earth. Okay, so about 2 billion years ago, the rise of oxygen and the rise of the eukaryotic cell. And the eukaryotic cell is a much higher energy, faster metabolizing cell. It requires something that's going to help drive that faster biological process, and oxygen has that capability. So that's why they came hand in hand. So here's just an example of a cyanobacteria. And the green look that you get from the chlorophyll that is involved with this photosynthetic process. And these are the little critters that gave the rise to oxygen and are why we are oxygen breathers today. Okay, so let's just talk about the sort of broad brush development of the eukaryotic cell. The top 3 cells that you see there going from left to right, you start with the ancestral cell, which is going to be a prokaryotic cell. It's got one membrane, it's got its DNA inside of it. And so with time, you might get some development of protection Again, this would be unequal reproductive success, and the fact that those which had these additional folds and eventually internal nuclear membrane were more protected, they were more concentrated, they had less competition, and they had much more success at continuing to reproduce themselves. So eventually you get this nuclear membrane. So that's step one, is maybe, how do you get the nuclear membrane? And this would be it. And then you would also get maybe even more complex folds around the nucleus. Okay, then B here, there's the idea that maybe for lunch one day, one of these ancestral cells wanted to eat some bacteria that was, say, very good at processing energy. And then it turned out that instead of destroying that little prokaryotic cell, that in fact they both found that they could live inside of each other much better. And so this was sort of what was referred to in very briefly in one of those space-time movies I showed in class, where it said maybe it was a lunch that went bad, which I thought was kind of a humorous way of saying it. But nonetheless, you develop this symbiotic relationship between now what are called mitochondrion and what was a prokaryotic cell. Now the thing is that there's other forms of these energy generating cells, and one of them is the chloroplast, which is responsible for photosynthesis. So if you were a mammal like ourselves, you only have the mitochondria, but if you were a plant and you were existing off of photosynthetic energy, then you could develop a symbiotic relationship also with the chloroplasts. And so that's believed why there's two chains there. Okay, now we're going to go back to the eras and the periods. And just so you know, and pardon me for flipping slides here, but I'm going to do it. Well, apparently I can't go backwards. So this is the eras and the periods that were for the last 550 million years. This is the last of the eons, okay, the visible life eon, as you will. And what we're going to talk about here, and the reason this is its own eon, and of course broken into 3 eras and multiple periods, is that a very special event happened on the Earth, happened once on the Earth, and that is something called the Cambrian Explosion of diversity. And this is a period in time. You have to understand that up to this point in time, all life forms were really basically in the ocean, and they were microscopic, and there may have been a few cells here and there put together, but things were very, very, very simple. And you developed the eukaryotic cell, and you had a faster metabolism happening because it was aerobic based, and you got to this period where things just took off exponentially, and all of a sudden now multiple life forms are filling different ecological niches throughout the oceans. Now again, we're still in the oceans, and the body plans and the complexity start to grow. And so you really start to get specialization for the first time on a large scale, and a diversification. So there are about 30 different body plans that are known in modern animals, and most of those 30, if not additionally more, but some have become extinct, plans were developed during this Cambrian explosion. Now this happened around 542 million years ago. Just 550 is good enough. And the period that it started was called the Cambrian Period. That's the Cambrian Explosion. And we really owe the diversity of life on the planet, and the fact that this points to a common ancestor, if you will, from all this diversification that happened in a very short period of, say, less than a few tens of millions of years. I think each tick now on the bottom is 10 million years. So in some period on the order of 20, 30 million years, we had this diversification. So we need to ask ourselves some questions because scientists are like children. They always ask questions. Why? Okay, I know that science doesn't answer why, but maybe we should replace it with how. Or what are the reasons that the Cambrian explosion occurred very suddenly and yet so long after the development of the eukaryotic cells, which started to happen around 2 billion years ago, okay, which means this Cambrian explosion is 1.5 billion years after the first eukaryotic cells developed. And then, what are the reasons that this large-scale diversification hasn't happened again? Why did it only happen once? So my next slide has a list of why then and why once, 4 reasons. Okay, why then, why once? Okay, why then? Well, number one was oxygen is very important for rapid metabolism and high energy metabolism. If you're going to diversify and have all these things have to happen on a fairly rapid time scale. And you have to have things pushing them and driving them. So aerobic energy was just not fast enough. So high intensive aerobic energy was required. So you couldn't have it before the rise of oxygen is basically the point of that one. And the reason why is because faster metabolism and aerobic metabolism were required for such an explosion to occur. Okay, now we also have the development, number 2, of genetic complexity. It could be that the complexity of the genes so that it could express itself in so many varied ways and specialize in so many varied ways took another 1.5 billion years from when the first eukaryotic cells developed. So you have to wait around some period of time for the genetic complexity required for this explosion to occur. So that would be another reason why it may have been 1.5 billion years after the rise of oxygen. Okay, and then why then as well? Well, we haven't talked about climatic evolution of the Earth too much. I remember we briefly touched on it previously, but it turned out that it was quite unstable, and then somewhere along the line of about 500, 600, 700, go back to, I don't know, a billion years ago, roughly, that the climate stabilized. And the stabilization probably was really important for the development of genetic complexity and for the survivability of all this life. And then now you've got the genetic complexity, now you've got the oxygen levels, and you've got stable environments, and poof, everybody's happy. Everybody starts moving into the different ecological niches, some deep, some shallow, some embedded in plants, some embedded in under rocks, whatever it took to find a place to have your unequal reproductive success and take off, and then of course evolve into a different direction unique to that environment in which you had taken a foothold. Now, the last one, 4, marked in red here, would give the reason of why only once. Okay, well, why only once? It's very possible that at the time, there just was no dominant predator in any of these ecological niches. So, you start to make a colony under a rock, there's not some other critter down there already who's just going to come along and eat you up. And so, we're talking about a bunch of microbes here, but even microbes could have been predatory at some level. But the idea is that everything was open. It was just, free land grab, if you will, free ocean bottom, ocean water grab. And so at that point in time, all these niches were open and there was nobody there already to take them or to come in and eat you up or do whatever. And so the lack of dominant predators, I don't mean T-Rexes, they didn't exist yet for another, 100, 200 million years. So we're talking about just basically these little microbial things and being in different ecological niches, and that they were open. There was nobody there already or nobody who could come along and wipe you out if you took one. So that would be the why only once, because guess what? Today, good luck finding an ecological niche that's wide open and some predator is not going to come along and see you and go, oh, great, yummy, and there you go. Okay, so if we move along to beyond the Cambrian Explosion, we have a period where we all obviously got plants and animals colonizing the land. So it turns out that first the plants colonize the land, and then later after that, you'll find that animals colonize the land. And then as animals colonize the land, you developed into this Permian period where you have a lot of huge reptile-ish type creatures. And then, of course, the dinosaurs come along around 250 million years ago. And they're sort of chicken-like and small, and they're very successful. They take over, they grow to be huge through a series of processes that drive that movement toward large creatures. They ebb and flow through the Triassic, they ebb and flow through the Jurassic, then they ebb and flow through the Cretaceous. And then somewhere at the end of the Cretaceous, about 65 million years ago, they seem to have just vanished from the planet, along with some huge percentage of the biomass. And this period of 65 million years ago, turns out there's a scar left on the Earth, if you will, a relic. And that relic is this thing that's called the KT boundary for the Cretaceous Tertiary. And don't ask me why the K and not the C for Cretaceous. Excuse me. So anyway, if you go around the world, you see things like you see in this image here, where you have this line, this layer, if you will, and there's this black soot layer. Wherever you find this, which covers the planet fairly ubiquitously, you find a couple things. First of all, there's a whole bunch of life forms, including dinosaurs, that you only find their fossils below this line, this boundary, and you never find it above. The other thing is that in the boundary layer itself, you find a tremendous amount of soot, and that this is believed to be due to the biomass of carbon products that were burned during some catastrophic global fire, if you will. And the other thing you find is in this soot is high concentrations of iridium. And that is very, very interesting because when the Earth formed, out of all these cosmic abundances of the elements on the periodic table, those which are further down the periodic table were heavier because they were more massive, and they would sink more toward the center of the Earth, and therefore they are rare on the surface of the Earth. Iridium, gold, platinum, there's plenty of that stuff on the planet. It's just not in the crust. It has, differentiated down into the mantle. So you don't find the cosmic level of abundances of iridium on the surface of the planet. But when you go and look in this KT boundary, you find cosmic level abundances of iridium, which would indicate that this iridium came from off-planet, and therefore this supports the idea that whatever caused this came from off-planet. So you got massive fires, you got massive extinction, and you've got chemical abundances which indicate some kind of foreign object. And this led to the idea that there was some kind of meteorite that struck the planet and caused mass extinctions. And believe it or not, it took until the 1990s to actually find the scientific evidence to demonstrate this. Moving on to the next slide here, we see that the KT impactor, if you will, is likely to have struck the Earth in the very northern area of the Yucatan Peninsula. So in our graph here to the side, you see a map, the inset map. You see the United States and Mexico. You see the red area, which is the area shown in the larger topographic map. And this you can see is at the very top of the Yucatan Peninsula. So if you ever go to Cancun, which is on the peninsula east coast there, then you can go around and search around for little pieces of meteorite that hit the Earth 65 million years ago and bring them home and put them on your shelf for a collection. This crater has ages that match the 65 million year old mark of the KT boundary. It's about 200 miles across, and models and simulations would suggest that was about 10 kilometers across, which puts it sort of in the every 100 million year regime for striking the Earth, and also makes it about the size of the city of Las Cruces, just for reference. Now, the force of this is believed to have been about equal to 100 million hydrogen bombs. And this is pretty much, if you will, a smoking gun for what would cause the mass extinctions 65 million years ago. And you can watch all kinds of cool things on the internet showing simulations of this. Here are some snapshots from one of those. Something coming into the Gulf of New Mexico at high speed, causing shock waves in the atmosphere as it hits, and then, of course, creating this tremendous crater, this tremendous explosion, and, of course, tremendous fires. And so the idea when you model this for the climatic evolution of the planet is first you get a lot of dust and smoke in the atmosphere, and you're getting soot from fires and things like that. Now, this is a major shutdown of photosynthesis, okay? You shut down the sunlight, then plants are starving for energy. They're going to start dying. So anything that feeds off a plant is going to be pretty much a goner as well. The next thing is that you're going to cool the earth tremendously because you're going to shut down the input energy that was used for the greenhouse effect, which kept the atmosphere warm. And so we know today that the greenhouse effect keeps the earth 35 degrees Celsius warmer than it would be if the greenhouse process did not operate. So the fact of the matter is we get a freezing of the planet, or at least a very, very cold period of the planet. Now, as things clear, you're eventually going to get a very warm phase of the planet. And part of that is because you had this tremendous release of carbon dioxide and water vapor into the atmosphere. Once all the soot moves out, now you've got the sunlight, but you have a very efficient greenhouse operating, and so you get basically an unusual warming. Okay, and so some of this unusual warming with some of the particulates that are still left in the atmosphere is going to drive some chemistry, which is pretty nasty, some nitrous oxide and things like that, that basically are poisonous gases and toxic. So again, immediate cooling followed by long-term warming, and then while you're in the long-term warming phase, toxic gases. So an unpleasant environment, and only small creatures seem to have survived. It turns out that the Earth has a great deal of extinctions, at least 5 major extinctions that we can identify. And so here we are going back to from about 400 or 500 million years back. So this process covers the present day, which is the bright purple at the very right, around 0 years in the past. The KT boundary moving to the left is at 65 million years ago in the past. You can see that the percent of extinctions was quite high, about 40%. And then you move back and you are in the late Triassic, which is between the yellow and the gold bands, and this was about 100 and 200 million years ago. This is the period when the dinosaurs actually started to arise. So there was a mass extinction that did leave the Earth open for the dinosaurs to command the Earth, which they did from about 200 million years ago to about 65 million years ago. Now, before that was what's called the Permian extinction, and that happened around 240 million years ago. And that was actually the largest mass extinction on the Earth. And then moving back in time, when life was much more simple, about the time that plants and animals had just colonized the land, we have the late Ordovician period and its mass extinction. So altogether, about 5 mass extinctions that we have following the Cambrian Explosion, which I will remind you is just off to the left of the graph there at 540 million years ago. Okay, let's talk about some other things that we don't want to have happen to our planet, which we don't really understand whether they naturally cycle on and off, or what happens with them. One of the questions in exam 2 was what causes the Aurora Borealis, which we're showing a beautiful picture of there on the right. And that left picture kind of shows the process. To remind you, you have charged particles coming in from the solar wind of the sun. And these particles come in at very high speed, 400 to 800 kilometers per second, and they get trapped in the magnetic field. They get trapped on the field lines that you see the images there of what those look like. And they get funneled, as you can see that one arrow funneling downward toward the North Pole. And they can actually then ride that magnetic field line all the way down to the surface of the Earth near the pole. And then as they collide into the upper atmosphere, they release light from that energy of that collision and cause the Aurora Borealis lights. Now, if the magnetic field did not exist, you take away all those lines that are surrounding the Earth that are protecting the Earth from the charged particles. All those charged particles now can slam directly into the atmosphere. That's going to cause some problems. It's going to break up oxygen molecules. It's going to kick some of them out into space. It's going to thin the atmosphere over a long period of time. It's going to change the chemistry of the upper atmosphere, especially the ozone layer. Now that ozone layer goes away. We're going to be bathed in ultraviolet light because that's sort of a layer of molecules which effectively absorb ultraviolet light from the sun very well. And then you've got the problem that the charged particles that are highly energetic can come all the way down to the surface of the Earth and strike your body. They're like little billiard balls. They come in, they knock apart your DNA, they cause all kinds of havoc, which turns into cancer. So we love our magnetic field. Let's keep our magnetic field. Another continuing danger, the threat of impact. So we have a lot of what are called NEOs, NEO for near Earth orbit asteroids, and we are being very vigilant about looking for these with telescopes. Unfortunately, due to the COVID-19, all those searches have been shut down. And in fact, most of astronomy is completely shut down, and we're not really watching the sky at all or doing any astronomical research at this period of time. Sort of an unprecedented situation. Now, impacts strike the Earth all the time. As we'll show in the next slide again, I'll show you the relative frequency of these impacts with impactor size. And the situation is that we have a lot of scars on the planet, but because we have a lot of weather and erosion, because we have liquid water on the surface and a rain, water, water vapor, liquid water cycle, we have erosion, and so the number counts the scars are wiped away with time. But we do have some counts, and so this image is showing you some of the counts that we have. And we go around the Earth and we count them, and we learn a lot about the relative sizes and ages of these impacts. But we get a lot more information about this by studying them on the moon, where you don't have erosion, and so the scars are basically kept there. Now, so this is a picture of the Tunguska event that happened in 1908, and this is 10 years before the Spanish flu, a pandemic that is something that we should all learn some lessons from to help us get through the COVID-19 pandemic. But it's sort of editorial comment. I hope you'll YouTube some of those videos to watch about what happened there and how it went in 3 phases and whatnot, so that you can be more educated and make better decisions, not just for yourself, but as a responsible citizen of the world. But in 1908, a large impactor, about 30 meters across, that's about 100 feet, so the size of a football field, exploded in the air in Siberia and caused a massive, if you will, flattening of the forests. It set them on fire, but these for 200 miles around, trees were knocked down radially outward from the center of where this explosion happened up in the atmosphere above the ground. And they snapped like toothpicks for 200 miles. So this was a major explosion in the atmosphere. Now, this kind of thing, a 30-meter across impactor, if we go down to our little frequency diagram in the lower left here, we can see 1 meter and 10 meter and 100 meter. So with 30 meters is somewhere in between there. Let's just say it's roughly halfway in between those two. We move upward on the graph and we see that Something like that strikes the Earth somewhere between every year to every century. Okay, so I would say that it's more likely that this is a once in a century type of size. You know, there's always some slop in the data um, because data are never perfect. But let's just say that this is the typical event that happens about once every 100 years. Well, and lo and behold, on St. Valentine's Day in 2013, roughly about 100 years later, Russia had a repeat of this. It wasn't quite as powerful as the Tunguska event. Now, below is a little video link that is only a few minutes long, showing you the power of that impact from different video cameras from these people in Russia. So I'm going to put that link on Canvas so that you can actually directly link it to it. It's actually a very telling little video, and there are, of course, documentaries on it if you're interested beyond what we're talking about here. Now let's wrap up this lecture with some discussion about human evolution. There's so much that has been learned in the last 20 years that I really can't do it justice. But I'm going to basically just tell you that when Darwin came out with this idea that life forms evolve from one form to another over time through isolation and specialization in different ecological niches. A lot of people went, bat poop crazy, thinking that this implied that humans had evolved, directly from chimpanzees and apes. And this, of course, is not true. What it really implies, if you let go of your irrational thinking, is that chimpanzees and gorillas and humans had a common ancestor. And we have a great deal of DNA overlap with chimpanzees, for example, about 98% of our DNA sequences. So only 2% difference between us and chimpanzees gives you the complete difference in the expression of these life forms between humans and chimpanzees. But we definitely had a common ancestor, and somewhere in the past, the two became isolated in their ecological environments and therefore became more specialized through unequal reproductive success and eventually looked less and less and like each other and developed special traits that were very different from each other. And that is the process that happened. It is natural. It is not directed by some, goal for higher intelligence, but somehow our process was that building a bigger brain, piling gray matter on top of our smaller brain, developing artwork, understanding symbolism through language, through art, and understanding a consequence of cause and effect over long periods of time, and developing long memory was highly successful for us to continue to reproduce in the ecological niche that we were filling. So that is the story of human evolution. And so here is basically the plan diagram showing you our common ancestral relationship and how far back in time we go with different versions of apes, monkeys, and prosimians, okay, if you will. Okay, major misconception. So you can see the artwork in the lower right corner. No, we did not evolve straight from gorillas. And if I put that on the test, I hope you'll get it right. Okay, now what is fascinating that has only been really learned in the last 20 years is that there were multiple species of hominids existing in parallel in different regions of the planet. So this development toward intelligence and the development towards the modern human was not a single lineage or a single line. It broke in also into multiple channels that simultaneously existed on the planet. Now, in fact, toward the present, as you can see, where there is, you can see we've got the Neanderthals and the Homo sapiens kind of existing in an overlap situation. There actually was, these people met each other. They hunted with each other. They crossbred with each other. In fact, today we have something like preserved 30% of the Neanderthal genome collectively in the modern Homo sapiens genome. It's a fascinating situation. But in the end, there was intelligence and competition, and it turned out that we must have been the most aggressive because apparently we clubbed to death any of these other competitors or somehow squeezed them out. They weren't able to compete with us, and we're the only hominid left standing. It would be fascinating if there was multiple hominids today, but one could argue that maybe the transition of our society into a modern civilization and the development of technologies might require total domination of the planet. And most of that being because why? Because then, just like in the early days when the RNA might have been isolated and not be competing to reproduce itself and with other agents that could compete with it or destroy it or what, we now also don't have other beings or other hominids competing for the environment that we have to protect our ecological niche from. We can take over the entire ecological niche, and from that point on, we can then start relaxing, intellectually developing art, intellectually developing language, intellectually developing technology, and bang, we have our modern civilization. And then maybe we can have a new explosion of diversity, which will happen in the next phase, which is when we go out into space. So we went from the oceans to the land. Now we can go from the planet itself out into space. That'll be a fantastic evolutionary trend. But again, what am I telling you here? It was not a single lineage. We did not split from the chimpanzees, say some 7 million years ago, and then it was a direct line to the modern Homo sapiens. No, there were multiple splittings. We're just figuring this out. We're just understanding how many there were and how varied they were and getting them dated. But it's a fantastic story, and somehow we are the only one that continued and didn't get snuffed out in that way. This next slide is another diagram which also is very similar to that, showing that humans evolved from over the last several millions of years, and that you can see as you vertically go upward on this diagram, that would be at a constant time in the past. You can say for example, around 2 million years ago, going from 0 at the left to 2 million years ago, that there were at least 3 different hominids that existed on the planet simultaneously, and then of course those became extinct. And so again, this is not a lineage that went from the upper right. These are branches that were shown in the previous diagram, and in fact from those branches you have the final standing Homo sapiens us. Now I would like to end with the last slide. Yay. Are we advancing beyond the Darwinian curve? And I'm going to say, spoiler alert, the answer I think is yes. And there are 3 reasons why. Number 1, we have cultural evolution. Cultural evolution involves the process we are involved in right now, which is previous generations conveying knowledge that's been developed from their previous generations to the next generation, which will then do the same. And we are doing that, and that process is accelerating dramatically. So one of the things we're doing is transmitting knowledge that's accumulative over the generations. This is unprecedented. The process at which it happens is very rapid. It's not to say that it doesn't happen in the animal kingdom, but in the animal kingdom it is very, 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 very slow process. Okay. The next thing is we've now developed technology. From the first civilizations that did agriculture onward to the modern day, rocketry and commercial airlines and computers and internet, we have continually developed technology. This also is accelerating faster and faster and faster. And there's a feedback loop on that, because as computers get more powerful, we can do things faster. which then allows us to do things faster again. So again, it is accelerating the knowledge for cultural and evolution. Finally, CRISPR, genetic evolution, being altered by people going in and engineering what's in your genes. Okay, there's so many examples of this that I can't go into them all. The first one is CRISPR. CRISPR is where you can go in and you edit a particular gene and you can actually change what that person's reproduction of their further cells is now going to have the new gene in it, and you basically have turned that person into somebody that might have, say, what this doctor in China did, where he gave these two twins some resistance to AIDS, so that they were not susceptible to the AIDS virus. And I've heard that they're using CRISPR to help people regain hearing, that they're using CRISPR to try to help people to regain eyesight. Okay, so this is very fantastic. The other one is that we've done situations where people have put together zygotes, or I should say fertilized eggs, and they go through the fertilized eggs, and let's say that both the parents have a gene that would provide a very mortal sickness to their children. Okay, and so there's a 1 in 2 chance that the zygote would get this gene, and so they basically go through and they find those that didn't have the gene, and so then they implant the female with the egg that fertilized egg that doesn't have those genes, and now they have actually stopped forevermore all future generations in that family from having that gene. They've gotten rid of it entirely. So that is another form of genetic evolution that we're seeing. And so these things are going to completely change the Darwinian curve. So unfortunately, a lot of this might get used for what we would call cosmetic reasons. That would be interesting. I'm sure there's going to be billions of dollar industry on that. Then you've got the idea that you could make super soldiers. You've got the idea you could make super athletes, or maybe super workers that are stronger, could live without water longer, so that they can work in the desert and build more cities out there, or expand the environments that humans could live in. It's endless, okay? And as soon as we figure out how to handle all this in an ethical way, I'm sure we will. Okay, well, that's going to end this slideshow. I hope you enjoyed it. I realized that I didn't say the magic word, and so I got to say the magic word. And gosh, this is ridiculous because it's at the end. And the other thing is that your intelligent professor here actually forgot what the magic word was. So you have to give me a second to say what the magic word was by looking it up. And unfortunately, I do not have a quick and dirty way to do that. Okay, so let me look at that real quick. My apologies. And I'm almost there. And the magic word is quasar. Okay, so there you have it. Okay, so anyway, then you know that you need to take the quiz, quiz C6, and you need to take the attendance quiz, which I just gave you the magic word for in this lecture. That would be AC6. Those will be due at basically 11:59 p.m. on Tuesday, the 31st of March. Okay, so don't forget to do those things, and then we will have no homework on this set because you already handed it in during our extended spring break. Okay, everybody, I hope that this worked, and I hope that you enjoyed this, and I will see you online. Take care.